Assalamu alaikum. How are you doing, everyone? So today I will show you how to do the staining, hematoxylin eosin staining. I will be very brief about the process because, as you know, the best thing is to do it by your hands. So this is just to give you an idea about the staining, about the processing, till you return back, inshallah, safe, so that you can do it by your hands. Now, for the setup of the staining, as you remember, this is called eosin, hematoxylin eosin staining. Means that we use two stains for this staining. Hematoxylin, and this is as a powder, okay? And eosin. Now, already you studied in the theory the different types of the hematoxylin and eosin. I will not going to mention these ones here. They are just mentioned in the theory, so just you can refer to your theory section to remember the types of these stains and how they are prepared, and also how we deal with them for the preparation. Already they are prepared here by Ms. Nabila. Okay, thank you, Ms. Nabila. So here you go. This is the stain. See, this is how it's appearing. This is hematoxylin, and this is the eosin. Okay, eosin stain. Now, you need to remember something very important. As we mention all the time, the labeling is very important. So here we have different steps, which is hematoxylin eosin staining protocol. And there is different steps here. As you can see, these steps, they are subcategorized into different categories. For example, here, rehydration, hematoxylin staining, bluing and washing, dehydration, eosin staining, dehydration, then clearing, and then mounting. If you look at these ones here, you can tell your sections already they are in paraffin. So you need to have this paraffin to be milled down a little bit so that it will be easy to remove it with the xylene. So here you need to warm it in the oven at 56 degrees. Then there is the removal of the wax. So you're bringing the sections to the xylene. Remember, they were in the paraffin sections. So you will remove the paraffin, and also they don't have water in them. So you need to rehydrate. That's why you do descending series of ethanol starting from 100% and going down 90, 70, some people they use 50, and then tap water. And this you need to change it so that to make it fresh. Here now you bring water to your sections. By this you will be ready to stain them in the hematoxylin, which is miscible with water. So you bring them to water, they will be ready to be stained with the hematoxylin. After the hematoxylin, you need to show the blue color of hematoxylin. Remember, the hematoxylin is stain of what? Acids or bases? You need to think about it. Hematoxylin staining the acidic moiety in the cells. What's the acidic moiety in the cells? They are the nucleus, the nucleic acids. So it will be staining the nucleus with blue color. That's why then we need to enhance this blue color by bluing step. So here you will be washing from the hematoxylin, and then you will be treating with what we call lithium carbonate. This is saturated solution, and it's here, it's already prepared. Okay, this is for bluing, enhancing the blue color of hematoxylin stain of the nucleus. So once we do the bluing, we wash. Remember, there is timing for each of these ones. I will tell you about the timing in a bit. Then after that, we need to move to stain with eosin. Eosin is staining the basic moiety of the cell. So it will be staining the granules, it will be staining the cytoplasm, and so on. So anything that is not acidic. Now eosin stains the cells that they don't have water. We don't want water to be there. So what we need to do, we need to remove the water from the sections again. So that's why we need to first incubate with ethanol 70%, then 90%. Then sometimes people, they add the 100% before the eosin. Then we do the staining with the eosin. After that, we continue dehydration. And then we bring the sections back into the xylene stage. Why we bring them back to the xylene? We already removed it from the beginning. Because when we are going to do the mounting medium that we use is the DBX. DBX mounting medium does not dissolve in water. So you need your sections to be the same medium, which is xylene can be dissolving in DBX. It's miscible with DBX. Okay, so each one of them has its own timing. This timing is optimized for each lab. So you need to optimize it by yourself, especially those ones for the stains. So for the hematoxylin and the eosin, sometimes when you receive a new stain, a new powder from the manufacturer, and then you store it for a long time, these stains might undergo either oxidation or reduction. This will affect the stability of the stain and the efficiency. So each time you prepare the stain, you need to optimize the time for staining. Let's go for the staining now. Okay, so now for the staining, you need to make sure that you have containers, and these containers, they are suitable for the staining reagents. Remember here, xylene, we mentioned xylene is reactive with the plastic, so we need special form of the plastic that won't be affected by the xylene. Also, for the stains, these ones just we place them to show you the color, but in reality, we need to protect these from light, so that's why we need to put them in also dark containers so that they won't be affected by light. This is very important. Now, for the beginning of the staining, these were sections. Already we bring them from the oven, okay? And they are ready for the xylene. We already started the initial one slide. So you need to make sure the timing is fixed. And I will move this one to the next one, okay? And these are the ones, so I will start here. I will put them in the xylene, and then I will start the timer based on the steps here. So xylene 1, 3 minutes, 2, 1 minute, 1 minute, and we continue. Okay, so I will continue up to the 70% ethanol, and then I will show you how we proceed with the staining. Okay, so here, this is the first step. I need you to see here. You compare the sections with paraffin, and now the paraffin has gone from the sections. No paraffin. Only what's remaining there is the tissue attached to the glass. Same here. No paraffin. Okay? Now we need to remember something. Don't allow or never allow your slides to dry while staining. So directly go to the next step. Never allow them to dry. Now we will continue. Okay, so now I bring the sections. I pass through these ones. And then the sections now, they are in 70% ethanol. I will be now bringing them. They are okay to be in distilled water. Now we need to remember, each time try just to remove this excess fluid, because with too much staining you will be contaminating the next containers. So try just to remove this excess as much as you can. Now they are in water, and then I will incubate them. Okay, so now after distilled water, I need to bring them to the hematoxylin stain. I need you to watch again. See the section now, only the tissue here, no paraffin, and it's in water environment. Remember, for the second time, I remind you why we bring them to water here. Hematoxylin is prepared in water, so it's miscible with water. Okay. Now we'll be incubating them with a stain. According to my protocol that's optimized already, we need to incubate them with hematoxylin for 10 minutes. Sometimes you might go to the places that they incubate for 6 minutes or 8 minutes or 5 minutes. This is depending on the hematoxylin, whether it's new or old or the preparation. Okay, so now our sections, they finished their hematoxylin staining timing. So I need to transfer them from hematoxylin to distilled water according to the staining protocol. Just I want you to see, I will just do one tab here to see what's the color of the hematoxylin before bluing. Now in water for 30 seconds. So again, we are here, hematoxylin, 10 minutes, tab water, 30 seconds, and then lithium bicarbonate for 30 seconds, which is bluing step, and then under tab water for 30 seconds as well. So now they should be done. I will just remove them from water. This is after the removal of the stain. Look at the section here, how it look like. This is the color. It's pale color. Now I will bring them to the lithium carbonate for the bluing for 30 seconds. Okay. Now this is a bluing step. It will enhance the blue color of the hematoxylin. Now, so after that one, I will show you the coloration change. Supposedly now there is 30 seconds have passed. I will put it here by tapping, and I will show you the color of the sections, how it became. Look here, this section at a white background. See, now it's blue in color. This is one section. This is the other section here as well. See, blue color. It's enhanced. You can retain back the video and compare the two colors. And this is the third section as well. Okay, so before my sections dry, I will put them again in distilled water. This step I will do it under the tap water, so that this water will be changed all the time. I will again, remember, just don't make the water flow on the slides, so that your sections won't be fall down, and here we go. Okay, so just I will return it to the sequence now. Remember, these water need to be changed each time, and then after that I will go to the, I will now remove the water, dehydration, and then we will go to the eosin. Remember, eosin is prepared in 95% ethanol. So in order to stain the sections with the eosin, you need to bring the sections back into ethanol. In other words, you need to do dehydration so that they will be stained with eosin. Okay? Now I remove them from 90% ethanol and then the eosin. You will see also, if you look at the blue color, it will be enhanced more after the water and the ethanol. I hope it will be clear here. See? Okay? Now let's continue to the eosin. Now, according to my protocol here, that's the one that's optimized, we need to incubate in eosin for 5 minutes. And then after that, I will remove the water, and then I will return back to you while they are in the xylene. So, okay. Yeah. Okay. So, now we are done from the eosin. Then we remove the water. Also, we bring them up to 100% ethanol. And then we continue to the xylene. Now I need to just remind you of something. The xylene here, the one that we used at the beginning, we can't use it here. Each one needs to stay at the step that's corresponding to it. Never mix up these boxes together. This is for the beginning, before the staining, and this is for the ending after the staining. Otherwise, there might be a carryover and cross-contamination. Now, with these ones, now they are in the xylene. I will show you the sections, how they look like, and then I will return them so that the smell of the xylene is really annoying. See here, on the slide, just on a white background after the eosin staining. Okay, so now these slides, they are ready for mounting. For the mounting, I will just upload to you another video. Thank you.